So far in chapter 14, we've seen monosaccharides, their anomers, their reactions. Then we looked at disaccharides and some of their linkage naming and also their hydrolysis reactions. So monosaccharides, disaccharides, the final thing we want to look at in this chapter is polysaccharides. And poly sounds like the same root of polymer, which indeed it is. So this is going to be many sugars in a polymer. So all linked together. And you can see these are just little tiny pieces of the polymers, but we've got some polymers happening here. So when we look at these long polymers, these are going to be the ways that we store sugars in living organisms. So in plants and animals, you need to store some glucose for when you aren't eating or when you aren't photosynthesizing. So polysaccharides, great for storage of sugars. In plants, these are the sugars that we think about eating. We call carbohydrate storage starch. And there's two different types of starch, amylose and amylopectin. Amylose and amylopectin. The amylose and amylopectin are both able to be digested. We actually have a third type of glucose polymer in plants, and that's cellulose, but that one we can't digest. So these are the starches, the things that a plant would use for food and the things that we eat when we eat plants. These are both digestible. This is going to be a straight chain polymer, and we'll see that in a second. And then our amylopectin is going to be what we call a branched chain polymer. So straight and branched. And then plants also have separately cellulose, which we can see down here, okay? The cellulose is going to give us rigidity. So like celery, when you taste some of those fibers, that's due to some cellulose that you're chomping on and eating. We think of that as fiber, and that helps us with digestion too, right? But just in a different way. So taking a look up here at the amylose, you can see that it's just repeating unit after repeating unit, and usually I think we have about 300 to 500 of those glucoses in a row, so not 4, but a lot more. And you can see some of these start with the O in the ring, and you can see that there's an acetal, and another acetal, and another acetal, and another acetal, and these would just continue on in either direction to make the chain much longer. If I look at these linkages, you can see that the rings are already numbered for you. So I always start with the acetal carbon first, and I can see that that's carbon number 1 and that it's angled down. Don't be confused by this little bend. It's just one way that we draw these links sometimes. There's not a carbon here. It's just the same as if it was a straight chain down to the oxygen. So because it's angled down, I would say that this is an alpha 1. And then it's connected over here to the 4. And so in amylose, the only type of connection that I see is alpha 1 to 4 connections or glycosidic bonds. Looking down here at the amylopectin, you can see it's straight, but then it has this random branch here. And so my straight chain polymers might look like this, and my branches might look like this. I start to get a little bit more complexity to the structure. I can still find those O's in the ring and find some links. This is an interesting one. There's the O in the ring, and see how I go O to carbon to O. So this is a little bit different than the other links we've seen. And there's more links down here. You can keep finding even more. So if I look at, say, this first link, this looks a lot like what's up here, right? It's a 1 connected to a 4, and it's an alpha orientation here. On this one, you might be thinking beta. It looks like it's floating a bit, but it's not. Let's look carefully. So start with the O and go to that anomeric carbon there. And then notice what direction the O is coming out of that acetal. It's going down. So this linkage here is going to be alpha as well because it's pointing down, and it's going from carbon 1 to carbon 6 in the next ring. Remember we said we could count outside of the ring for that counting? So in this ring, it's carbon 1, and this ring, it's carbon 6. So I have two different types of links in amylopectin, an alpha 1 to 4 and an alpha 1 to 6. there is yet another type of link here in cellulose, and I'm going to let you look at that one. We don't have the enzymes to digest this particular link, so all of these are built out of glucose. Everything on this page is all glucose, and we can break down all of it except for this one. So this one we cannot digest. We don't have the right enzymes to break the bonds. So you want to think about labeling this link here. I'll leave that for you to do or for us to practice together. And then we want to just talk briefly as we wrap up on this page about animals and how they store glucose. So we, for example, store glucose in places that we might need it a lot. In our brains and in our muscles are two main places you want to have ready, immediate access to glucose so that you can quickly think or you can quickly move. So when animals store glucose, they store it in a giant glycogen polymer. And glycogen is down here. Looking at it, can you tell if it's straight chain or branched? Hopefully it's pretty obvious that this is branched, and in color it's a pretty awesome structure. I think you could also probably Google this or search it on MallView and be able to see this in a little bit more detail. This is a close-up of the glycogen over here, and you can see how there's a straight chain going through and then branches off of it. And you could, again, find some of those links and number them and say what kind of links we had in the glycogen. One last thing with the polymers, you can barely see it here because it's fuzzy, but you might notice that there's these brackets here and there's brackets here as well. When we note a polymer, something that's maybe 300 to 500 units long, or we're closer to thousands on these other two here, a unit repeats over and over again. And so this little n is going to represent the number of repeating units, which is to say that this little chunk repeats and repeats and repeats and repeats hundreds or thousands of times. So those are polysaccharides. They're longer, they're more complex, we have some different linkages happening, and they are ultimately still built from monosaccharides, like glucose is the one on this page, but they're just much, much larger. You saw actually starch in some of your lab experiments, and the starch, remember, was a huge molecule that couldn't move across membranes. So all of these starches need to be hydrolyzed, broken into smaller pieces before you can use them. Otherwise, they're stuck inside of your digestive system or stuck inside of a cell or something. So you want to be able to break them apart so you can move them around and use them. So that wraps up our carbohydrates. Now you'll want to practice a lot in your workbook and in class, and you'll be good to go.